Hi everyone, my name is Justin Wong, and this summer I performed a literature search and data analysis on treatments for recurrent pituitary tumors as part of a larger review. So just gonna zoom in here on the intro and go right into things. Okay, pituitary tumors are the second most common primary brain tumor, with an estimated prevalence of 80 to 100 cases per 100,000. And the primary treatment for pituitary tumors is transsphenoidal surgery. So surgery where they go from the nose to the center of the brain where the pituitary is. Or in the case of prolactin secreting tumors, those respond well to a type of drug called dopamine agonists. Unfortunately, there's a subset of tumors estimated to be approximately 10% of pituitary tumors that are classified as recurrent pituitary tumors, and these are invasive, so they penetrate surrounding tissues, and they also have unusually rapid growth rates and resist standard treatments. These tumors are primarily treated by the chemotherapeutic agent temozolomide. However, temozolomide has only been found to have a 37.5% remission rate, of which 30% recur. So there's an urgent need to develop more effective treatments for this challenging condition. For this review, I reviewed the efficacy of the dopamine agonists bromocriptine, quinagolide, and cabergoline, both as monotherapies and in combination with other drugs. And I also looked at the results of studies on developing treatments and treatments primarily used for other cancers, and reviewed their potential to treat recurrent pituitary tumors. I had 3 strategies for finding papers. I read through previous literature reviews and found the papers that they used. I did keyword searches on PubMed and Google Scholar, and I also did a cited reference search in Web of Science to find sources that cited the sources that I'd already found, to see if there had been any more recent papers. With these strategies, I found 257 studies using these drugs to treat pituitary tumors, of which 75 included patients with recurrent pituitary tumors, and 11 of these didn't separate results between those who resisted previous treatments and those who haven't been treated before. So I ended up using 64 studies for further analyses. Here you can see some of my results on the efficacy of dopamine agonists against recurrent pituitary tumors. The top left graph shows results against corticotroph tumors, so tumors of pro-opiomelanocortin secreting cells, which stimulate cortisol production and lead to hypercortisolism. Cabergoline is the only dopamine agonist that's been used to treat refractory corticotroph adenomas, and it's had success as a monotherapy, normalizing cortisol levels in 37 and 60% of surgery and radiotherapy resistant tumors, but it's generally been even more effective in combination with other drugs such as ketoconazole and pasireotide. The top right graph shows results for prolactinomas, against which dopamine agonists have been most effective. Against these tumors, cabergoline has been the most effective, followed by quinagolide, followed by bromocriptine, which has not been found to be effective alone. On the bottom left, we see results for growth hormone secreting pituitary tumors. Quinagolide has had the highest rate of growth hormone normalization, although it's only been used in 1 study with 5 patients. And both bromocriptine and cabergoline have had success when added to octreotide or lanreotide treatment. Finally, on the bottom right, you can see the results for non-functional pituitary adenomas. And similar to corticotroph tumors, these tumors have mostly been treated with cabergoline, though 1 study treating patients with either cabergoline or bromocriptine found a similar rate of around 30% significant tumor reduction. So overall, dopamine agonists have been shown to be capable of treating refractory pituitary tumors, and cabergoline has generally been most effective, and combination therapies where dopamine agonists are added to standard therapies have generally been more effective than dopamine agonist monotherapies. As for other treatments, the EGFR inhibitors gefitinib and lapatinib, which are mainly used to treat lung and breast cancer respectively, have been shown to significantly suppress hormone levels and cell proliferation, but they've yet to demonstrate hormone normalization or significant tumor reduction. And they also haven't been tested yet on refractory pituitary tumors, though they may still be effective against tumors resistant to other treatments. More encouragingly, the 9-cis and all trans isomers of the vitamin A derivative retinoic acid and the CDK inhibitor roscovatine have both demonstrated promising results in preclinical studies and clinical trials, though more studies with more patients will have to be conducted to confirm their efficacy. So that's that for my mini literature review. And this summer, I also worked on a project characterizing the expression of PE2F1, which is a potential target for Cushing's disease. So Cushing's disease is a rare endocrine disorder caused by corticotroph tumors, which secrete POMC, or pro-opiomelanocortin, which stimulates cortisol overproduction, leading to protein wasting, rapid weight gain, cardiovascular complications, immunodeficiency, and psychological disturbances. The primary treatment for Cushing's disease is transsphenoidal surgery, but recurrence occurs in up to 47% of patients, and current medical therapies, which target cortisol synthesis or symptoms of hypercortisolism, have had low efficacy and poor tolerance. So our group previously searched for a drug target that could more directly control cortisol overproduction by controlling POMC production. We found that this protein called E2F1 binds the human POMC promoter and that its depletion decreases POMC expression. We also found that E2F1 expression is cytoplasmic, which raised the question of how E2F1 enters the nucleus to affect POMC transcription. We also found that serine 337 phosphorylation enhanced E2F1 POMC binding, so we hypothesized that phosphorylation on serine 337 allows E2F1 to enter the nucleus and activate POMC transcription. To test this hypothesis, I immunostained sections from 3 normal human adult pituitaries and 4 Cushing's disease tumors. So in other words, I basically attached primary antibodies to specific target proteins, and then I attached secondary antibodies, which have a fluorescent molecule attached to them, to those primary antibodies in order to color each target protein with a different fluorescent color. Then I used a confocal microscope to image the sides, and I used ImageJ to create line scans, which graph the relative intensity of the signals in a line drawn across the cell. So here you can see my results. I confirmed that E2F1 is cytoplasmic and specific to POMC secreting cells. As you can see in the top row, E2F1 is only expressed in this cell that also expresses POMC. And in normal human adult pituitaries, PS337 E2F1 is perinuclear, so it surrounds the nucleus, which is stained by DAPI. And you can also see that in this line scan on the right, the PS337 E2F1 is mostly expressed just outside of the nucleus and just outside of the nuclear lamina. On the other hand, in Cushing's disease pituitary tumors, P2F1 expression overlaps with DAPI expression, and it's contained within the nuclear lamina. You can see that in these images here, as well as in this graph. So these results indicate that E2F1 may depend on serine 337 phosphorylation to enter the nucleus and activate POMC transcription. And molecules that block serine 337 phosphorylation may be able to treat Cushing's disease more effectively than current medical treatments. That's it for my talk. Thank you for listening. Are there any questions? I can switch back to the other poster if you'd like.
Justin, I had a question on this poster. All your work is very nice. So in Cushing's, P2EF ETOF1 is mislocalized?
What do you mean by mislocalized?
It doesn't follow the same immunofluorescence pattern as in a wild type person, right?
Yeah, that's what we found. We found
Yeah,
that
so
ETOF1 is specific to the POMC producing cells, but PE2F1 is seen in both cells that do and don't produce POMC.
okay, I guess my question is, in patients with Cushing's, are there any mutations in the gene encoding P2EF1 that might affect its subcellular localization or its serine phosphorylation?
I'm not sure about that. I haven't looked into it.
Okay.
But that would be interesting to look into, yeah.
And then I had another question about the dopamine studies. In the recurrent tumors that are susceptible, were those treated with dopamine in the first treatment? Does that make sense?
Could you repeat that?
Yeah, so if a patient is treated with dopamine in the initial treatment with a dopamine agonist, and then the tumor comes back, can they use dopamine agonists again, or is the tumor now essentially resistant to those compounds?
I think if it comes back, most studies just how do I say this? They would say that the tumor is resistant to it, and they would try a different treatment.
Okay.
Yeah, but yeah.
Thank you.
Any other questions for Justin? Well, thank you very much.
Thanks.